Hi, my name is Joanna Duong, and for my final presentation, I'd like to focus on examining the effect of Epstein-Barr virus on Hodgkin's lymphoma, a type of cancer. In this presentation, I will provide you the background of these two diseases, Hodgkin's lymphoma and Epstein-Barr virus. I will explain their connection and how it is important in an evolutionary perspective using the paper I chose. To start off, lymphoma is the cancer of the lymphatic system. The lymphatic system is a very important system in our body that has many functions such as removing interstitial fluid from tissues and fat absorption. It's also very important as it's part of our immune system. We have lymph, which is a clear fluid that contains waste products such as bacterial and viral products that become filtered by the lymph nodes. Lymph nodes contain lymphocytes such as B cells and T cells that are part of our innate and adaptive immune system, which we learned in class. So when cancers such as lymphoma arise, it becomes really dangerous because of how valuable our immune system is. Lymphoma can be classified into two different types, and that is Hodgkin's lymphoma and non-Hodgkin's lymphoma. This is based on the presence of Reed-Sternberg cells, and that's just abnormal B cells. Another difference is Hodgkin's lymphoma is very treatable and has a high survival rate in patients with proper treatment, whereas non-Hodgkin's lymphoma is a mixed bag of different cancers with variable survival rates and impacts. On the left is a picture of my dad, who actually had a non-Hodgkin's lymphoma called mantle cell lymphoma, and that was a very rare aggressive cancer, and he unfortunately passed away from it this year. So out of the two types, Hodgkin's lymphoma is definitely the less aggressive and the lesser of two evils. So when patients have Hodgkin's lymphoma, the symptoms they experience are swelling in the lymph nodes located in the neck and armpits, and that's due to the buildup of cancer cells. Patients will also experience a lack of appetite that leads to quick weight loss, fatigue, and night sweats. Cancers are more common in men than they are in women, and it largely affects young adults and old people. But luckily, there is a very high survival rate of 87% in patients for at least 5 years. The risk factors for Hodgkin's lymphoma is family history. So if someone in your family has had the disease, if you had HIV or AIDS in the past, and the most important factor is if you'd have Epstein-Barr virus infection. So Epstein-Barr virus is one of the most common viruses to humans in the world, and it is a human herpes virus. This is a double-stranded DNA virus that can be transmitted by the saliva, blood, or semen. The virus targets many cells, but particularly B cells and epithelial cells. That makes it important because uncontrolled B cell growth can lead to certain types of different cancers. This virus can have lifelong latency in cells even after infection and recovery because it resides in the nucleus and only expresses some genes. So it's a very common virus in the world that 90% of people in the world have had, such as you or me probably. And the scenario is that you've most likely had it as a child, but you didn't notice any symptoms. If you do have symptoms, what you'll have is mononucleosis, which is kissing disease, and that's mono. And these symptoms are fevers, swollen lymph nodes, and fatigue. In some cases, you might have chronic EBV or chronic fatigue syndrome, which is you get tired for your whole life for certain periods of times, and you might experience memory loss or confusion. What contributes to a high infection rate is that there's also no vaccine to EBV, and that's why we've all probably had it. So for the evolutionary significance, I like to focus on how EBV is strongly associated with many diseases such as Hodgkin's lymphoma, leading to half of the cases of lymphoma being caused by EBV. There's also other diseases I listed that EBV is associated with, such as gastric cancer, rheumatoid arthritis, Alice in Wonderland syndrome, and multiple sclerosis. So how it works is there's a latency of the virus, which can be seen in a diagram, and specifically in B cells, is that the virus will enter B cells and become integrated in the host genome and act as a provirus. This means that it will remain in the nucleus and only express a certain set of genes for latency and survival of the host cell. With the expression of these specific latency genes, this can lead to B-cell-derived lymphoma, and you can see that in the lymphoblastic lines. So EBV can be classified into two different genotypes. Type 1 is predominant in America, China, Europe, and Southeast Asia, whereas type 2 is mostly seen in Africa. As I said before, there's a specific set of proteins that are expressed when the virus is latent, and this is seen in the table on the right. EBNA stands for Epstein-Barr virus, Epstein-Barr nucleus antigen, and LMP stands for latent member protein. The one I want to highlight is in red, and that is the protein responsible for causing Hodgkin lymphoma, LMP1-2. And this protein activity is crucial in the pathways that will downregulate tumor suppressor proteins, causing the uncontrolled cancer cell growth. As you can see in the table, a lot of research has been done to understand the genetic background and basis of each EBV protein to diseases, but there's not enough research on how that's understood on the whole with the genome, which is why I chose this specific research article on the next slide. The article I chose was a study that did an evolutionary analysis on the EBV genome from countries such as Argentina. As mentioned in the class, there are many issues with genetic studies being skewed by having it skewed more on European descent populations and being underrepresented in populations of color such as Hispanic populations and indigenous people. So this study specifically chose to look at the geographic location of South America, specifically Argentina, by testing patients who've had EBV and may have also had Hodgkin's lymphoma or other diseases to see how genetic tests compare. They then took this data and compared it to different genetic analyses done on EBV that extended back to 1963 to see how the virus evolved. The photo on the right shows a phylogenetic tree of the EBV genome from different geographic regions, now including the new study done on Argentina, which is great. So what this study found was information of a high bootstrap value formed from the EBV type 2 sequence, and comparison found that there was a high segregation potential with Asian sequences from Argentinian sequences. This tells us that there was a bottleneck event which was highly likely, and it's similar to the development of other herpes human viruses. The evolutionary rate they found that showed a strong coevolution between the viral genome to the host genome. But the genes for structural proteins and genes that coded for enzymes showed a low evolutionary rate. But genes that coded for the viral immune escape had high variance, which led to there being a lifelong latency of the EBV virus in host cells. Overall, the data of the study showed us how the EBV genome worked and how it differentiated among different geographic locations along with its viral variability. There has to be more research to be done on how EBV connects to lymphoma, and there are certainly limitations to the study, but overall it provided us with a greater understanding of how the EBV genome worked. So that was my final presentation. Thank you so much for watching, and I hope you learned.